Okay, I have a few practice questions, a little bit of review here for Unit 2, Tissues and Skin. And again, most of these will just be some example questions that we can kind of talk through to see how you should approach questions about this material. And most of this is going to relate more to the physiology test than the anatomy test. Don't forget to use the practice activities that are posted for you in the Unit 2 Physiology folder and the Unit 2 Anatomy folder. And also don't forget that for the physiology exam, primarily, go back and practice again with your Mastering A&P homework questions. Even if you've already submitted those assignments, you can go back and use those questions for practice. And again, the idea is not to just memorize the answers to those questions, but make sure you understand why the right answers are the right answers, because that's what's going to help you prepare for the actual exam. All right, so just a little quick review here on epithelial tissues. Remember, epithelial tissues, where do you find those? They're mainly, they line hollow spaces inside the body. They provide coverings like at the surface of the skin. And epithelial tissues are classified in two ways according to how many layers of cells you have on that epithelial tissue. If it's just one layer of cells, like you see here, that's a simple epithelial tissue. If you have multiple layers, that's called stratified. And then you also classify those tissues according to the shapes of the cells. So if they're flat cells, those are called squamous cells. If they're kind of cube-shaped, cuboidal, and if they're columnar-shaped, they're called columnar cells. I know they don't really look like little wooden cutouts like you see here, but those are just some graphics from your textbook trying to give you an idea about how some of these cells are shaped. And then as you're studying the epithelial tissues, remember to go back and review, some example locations where these tissues occur, and think about function as well. Where would you want a simple squamous epithelial tissue? You would have one layer of cells, and those cells are very flat. So places where you need just a very thin, delicate lining or covering. And a good example, the little tiny air sacs in your lungs where oxygen gas and carbon dioxide are exchanged with the blood. That's a great example. Or when you guys get to Biology 202, you'll learn that the tiniest blood vessels in the body, the capillaries, are composed of walls that are simply a single layer of simple squamous epithelial tissue as well. And that's because those little tiny capillaries have to exchange fluids and gases and nutrients and wastes with the surrounding tissues. Okay, then in your connective tissues, remember this is a generic diagram of a connective type of tissue, and this is supposed to represent areal or loose connective tissue. But the key thing here is, when you're thinking about connective tissues, most connective tissues have the components in them that you see here, and so you want to be able to recognize these components and know what their jobs are. So remember, in connective tissues, you have the cells are more scattered around. In an epithelial tissue, they're kind of jam-packed together, but in the connective tissue, the cells are kind of scattered. And in most connective tissues, not all, but in most, the main cell type is called a fibroblast, a little purple thing that you see there, and there's another one over here. There are some other cell types that you see here, like your fat cells or your adipocytes. Those, of course, store fat. The macrophages, those are, we've talked about those. Those are like the vacuum cleaners that gobble up bacteria and fungi and viruses and dead cells of your own and damaged tissue components and so forth. Their job is to ingest and digest those types of things to clear them out. And then your lymphocytes here, neutrophil, mast cells. Mast cells help stimulate inflammation, like if you have a injury or a burn or an infection going on, and your neutrophils and your lymphocytes are, along with your macrophages, are involved with helping to fight infections. They're part of your immune system that you'll learn more about when you get to Biology 202, and also in microbiology if you haven't had that class yet. All right, then remember that within a connective tissue, everything that's outside of the cells, everything that you see here that I'm kind of coloring in, is called the extracellular matrix, and that's going to be composed of a jelly-like fluid that consists of lots of water and then all of the solutes that are dissolved in it. And that's called the ground substance. That's kind of a weird name, and that's what you're seeing here in kind of the blue shaded areas. And then connective tissues have fibers in them. Fibers are built from proteins, and they include the big purple ones here are collagen fibers. In orange on this diagram, those are your elastic fibers. And then finally, you have in blue, kind of forming this netting that you see here, those are called reticular fibers. So you want to go back and review the functions of those fibers. What is the job of the collagen fibers? What is the job of the elastic fibers? The reticular fibers more or less kind of provide a framework within these tissues. All right, how about an example question here? Which of the following tissues is composed of multiple layers of cube-shaped cells? Okay, so you just have to learn your terminology. Multiple layers, that's stratified. Cube-shaped cells are cuboidal, so that would be a stratified cuboidal epithelium if you were investigating a body area where that type of tissue takes place. All right, here's another one. Remember to be able to recognize where epithelial tissues occur in the body. Select each location that consists of epithelial tissue. All right, let's talk about these. Let me see if I can get my pen to work over here. Sometimes this works and sometimes it doesn't. So we'll see if it's going to cooperate. All right, epithelial tissue. What about epidermis? Remember with these questions, whenever you have a select all that apply, these are a good rehearsal for those of you planning to go on to nursing school. They have lots of these types of questions in nursing school. And you just kind of want to go down the list and treat each one almost like it's a true false. Epithelial tissue in the epidermis. Absolutely, yes. How about the dermis? Hopefully you're saying no. The dermis is composed of different types of connective tissue. So a no on that one. Air sacs, alveoli in the lungs. Yes, we were just talking about those. Those are composed of simple squamous epithelium. Osseous tissues, where do we find those? In the bones. Osseous tissues are actually connective. That's a type of connective tissue. How about the skeletal muscles? No, skeletal muscles are composed of skeletal muscle tissue. Review your different tissue types. Inside lining of blood vessels. Yes, we just talked about those. Any place you have an inner lining, you're going to have epithelial tissue at that location. Inside lining of the jejunum. Do you remember where the jejunum is located? That's that little second section of the small intestine. And again, yes, you have an inner lining in there, and that's going to be composed of an epithelial tissue. Oops, hang on. Okay, answers A, C, F, and G. All right, how about this one? Which tissue is often located at places where substances such as gases have to diffuse from one location to another with ease? Well, I was just mouthing off a couple of slides ago, so I think I already gave away this answer. Any place where you're going to have a lot of diffusion, you got to move substances from one location, one compartment to another. It's going to be good to have a very thin layer of tissue at that location. So which one of these would represent the thinnest layering? And hopefully you're answering simple squamous epithelium. Associate those with locations where diffusion of substances will have to take place, moving from one location or compartment to another. What tissue occurs in body structures that have to resist great amounts of tension in one direction? What kind of tissue? Tension in one direction, like if the tissue was getting pulled, but only in one direction, not multiple directions. And so in one direction, now first of all, what tissue component, what component of a connective tissue provides resistance to pulling? That's also called tensile strength. So you should know that. And those are your collagen fibers. All right, and those collagen fibers are arranged in different orientations, in different ways, in different types of tissue. So if your force is generally just in one direction, all those fibers are going to be oriented together in the same direction. And if you recall, that's your dense regular connective tissue. Those fibers are packed very densely together, and the tissue is classified as a regular connective tissue because the fibers are all oriented in the same direction. Where are some examples of where we would have that type of tissue? Tendons and ligaments are the primary examples of where you have dense regular connective tissue. Those are structures that connect bones to bones or muscles to bones, and they have to resist pulling generally in a single direction. Okay, which tissue occurs in the walls of many hollow organs and allows them to contract when needed? All right, so again, what we're talking about here, we said inside the wall of a hollow organ. Imagine something like a blood vessel or the small intestine. I'm trying to draw this in 3D here. Imagine you've got a tube-like structure here. And again, this could be part of the small intestine. This could be a blood vessel if we were looking at something smaller. So here's the inside, which is called the lumen, the inside of this organ or blood vessel. So what we're talking about is in here, inside the wall. If you cut this open and you're looking inside the wall in there, what kind of tissue inside that wall is going to allow that organ or structure to contract when needed? Anytime you hear contraction, think muscle tissue. So that's going to narrow us down to B and C. And so is it going to be skeletal or smooth muscle? Well, think about voluntary versus involuntary. Your skeletal muscle tissues are generally voluntary. Those are you think about it when you want to move a body part, skeletal muscles are contracting to help you do that. Smooth muscle tissues exist in the walls of hollow organs, and they're involuntary. You're not thinking about whether the wall of your small intestine needs to contract to help propel food that's being digested through your intestinal tract. You don't have to think about whether or not a blood vessel is going to constrict or dilate. So smooth muscle tissues exist in those locations to make those things happen. Okay, which of the following tissues would function best at surfaces or linings where frequent friction or damage occurs but moisture is needed? So let's think about this one. All right, so we know it's going to be an epithelial tissue because we're talking about a surface or a lining. Frequent friction or damage, like the skin, your skin is constantly bumping up into something. Or think about like the inside of your mouth. Your teeth are constantly rubbing up against the insides of your cheeks. You swallow things, particles of food that tend to rub up against those tissues and can slough them off pretty easily. So we want these tissues to be pretty resilient. But we also want them to be moist. So that's going to kind of help us narrow things down. All right, how about keratinized stratified squamous epithelium? Where do we find that? Hopefully you're answering the epidermis of the skin. But notice the question says moisture is needed. Your skin, the keratinization of the skin keeps the skin. That's a waterproofing substance. So that keeps the skin dry. That's not going to generally be a moist surface. So that's going to help us rule that out. Simple cuboidal epithelium. Simple means one layer. Tissues, an epithelial lining that's just one layer of cells is going to be very delicate. That's not going to be something you want at a location where lots of friction or damage would occur. Smooth muscle tissue, we just talked about that. That's involved in contraction of hollow organs, blood vessels, and so forth. So our answer here is D, non-keratinized stratified squamous epithelium. And that's the type of tissues you have like inside the mouth, back in your pharynx and so forth. Kind of like skin, but it's not keratinized because you would not want a dry surface in those locations. All right, body fluids. Certainly go back and review the information that we've learned about with regard to body fluids. Remember, the fluids in your body are divided up into extracellular fluids. Those are fluids that exist outside of cells in your body. And then you have your intracellular fluids. Those are fluids that are inside your cells. And most of the fluid in your body is actually inside your cells, but nevertheless, you do have fluids that bathe your cells, that exists in between your cells in an extracellular matrix. And another term for that is interstitial fluid. You also have fluid in your blood that's called blood plasma. Lymph is fluid that's located in your lymphatic vessels and your lymph nodes. And so just go back and review how fluids move between compartments. So it moves through, like out of the blood. The blood pressure squeezes fluid out of the blood vessels and into the tissues. And then what's the main thing the main force involved with water movement from extracellular fluid, your interstitial fluids surrounding cells, to the inside of cells, or vice versa? So hopefully you're answering osmosis. Osmosis is the main thing that dictates the movement of water from outside cells to inside, or vice versa. All right, so go back and review that information about body fluid. Body fluids come up all the time in A&P, so you need to go ahead and commit that to memory. Now, also, for those of you going on a nursing school, body fluids, that is a huge, huge topic in nursing school. All right, so let's just see. Let's do a little review on body fluids. Which of the following locations contains ECF? What does ECF stand for? Extracellular fluid. All right, how about A, the extracellular matrix of the dermis? Absolutely. Now, the ECF that's there kind of has more of a jelly-like consistency because there are so many solutes dissolved in it, but it's still classified as an ECF. There's lots of water in it. It is a fluid. How about a neuron? Where would you find a neuron? A neuron is a cell type in nervous tissue. So the cells inside a the fluids inside a neuron are not going to be classified as ECF. Those are going to be ICF, or intracellular fluids. How about blood vessels? Yes, we have ECF there. Lymphatic vessels? Yes. Smooth muscle cells? No, that's going to be ICF. How about the vitreous of the eyes? Remember that one? That's the jelly-like filling in your eyeballs. And yes, that is a place where you have extracellular fluid. The left pleural cavity, remember your pleural fluid that we learned about in Unit 1? Yes, that is existing outside of cells, so that's a type of extracellular fluid. How about the lumen, the interior space of the colon? Yes, you have fluid in there. It's outside of cells, and so that is also going to be a type of ECF as well. All right, by which method does fluid move from the interstitial spaces of the reticular sublayer of the dermis into a fibroblast? So lots of terminology in every unit in anatomy and physiology. So let's go through here and talk about this terminology. All right, interstitial spaces, those are the areas outside of cells within a tissue, the spaces that are surrounding the cells. The reticular sublayer of the dermis. Remember, your dermis is divided up into two sublayers. You've got the reticular sublayer, that's up at the top where you have the papillae of the dermis, the little hills. Oh, I'm sorry. Nix that. The papillary sublayer is where you have the papillae in the dermis. The reticular sublayer is the thicker part of the dermis down below that. Sorry about that. And anyway, so within the dermis, the main cell type is called the fibroblasts. These are connective tissues. Most of your connective tissues are dominated by fibroblasts. That's going to be your main cell type. They're called that because they are the cells that manufacture the fibers. They make the collagen, the elastic fibers, the reticular fibers. Those don't just magically appear from nowhere. They have to be produced or manufactured, and the fibroblasts make those and secrete them into the surrounding tissues. All right, so what's the main method by which fluid is going to move from the interstitial spaces, extracellular fluid, into a fibroblast? Is it going to be osmosis, filtration, hydrostatic pressure, facilitated diffusion? Hopefully you're answering osmosis. Osmosis is the movement or diffusion of water across a selectively permeable membrane, and in biology those selectively permeable membranes are generally cell membranes. Filtration is a process. You'll learn more about filtration when you get to Biology 202. That is where you have a lining that has pores in it, and fluids get squeezed through the pores or slits between cells. That's filtration. Same thing happens in your capillaries as well. Little tiny blood vessels, they do have little slits or cracks between the individual cells in the wall, and when fluid gets squeezed in between those cracks, that's called filtration. Hydrostatic pressure is like blood pressure squeezing fluid through those capillary walls. Facilitated diffusion, hopefully you remember from Unit 1, that is a process by which some solute moves into a cell or out of a cell by using a protein channel or carrier that's sitting in the membrane. So it's diffusing, but it needs help to accomplish that. All right, cartilage review. Cartilage review. All right, so this is a picture from your textbook of hyaline cartilage. That's the main cartilage type in the body, where you have, like on the ends of your long bones, that's an example location for hyaline cartilage. And you also have hyaline type cartilage on your ribs. You're going to learn more about cartilage in Unit 3. And cartilage has kind of a semi-solid extracellular matrix that's been stained purple in this diagram. And there are scattered cells around that are called chondrocytes. And those chondrocytes live inside little chambers. Each of those is called a lacuna. And so like there's a cell right there in white. That little pink spot is the nucleus. And so those cells, they live inside those little chambers, and they're responsible for producing all of the materials that make up this semi-solid matrix. Other locations for cartilage, you have elastic cartilage, the pinna, the outer part of the ear. It's elastic so it can rebound when you fold it over. You've got fibrocartilage in the discs between your vertebrae along your backbone. All right, tissue layers. Just a reminder about the major tissue layers that exist in the body and also within the skin. So got a typical kind of skin chunk here, but you should start developing a visual for, like, if you go from the surface of the skin down deeper and deeper and deeper into the body, if you cut open the skin, like if you stuck a scalpel in there, what are you going to pass through along the way, and what kinds of tissues are those? What are they composed of? All right, so if we start up here at the surface of the skin, as you guys know, consists of two major layers. The top one is called the epidermis. The next one is the dermis. So what kind of tissue is the epidermis composed of? It's stratified squamous epithelium. And then you move down into the dermis. Remember, with the dermis, we have up here where you have these little hilly areas poking up into the epidermis. That's your papillary sublayer. Then you have the reticular sublayer is deep to that. The papillary sublayer up here at the top is composed of areolar loose connective tissue. If you go down into the reticular sublayer, the bulk of the dermis down in here, that's where you have dense, irregular connective tissue, so lots of protein fibers, but those protein fibers are oriented in lots of different directions, and that's because your dermis has to resist pulling in many different directions. Otherwise, you'd just be able to rip little chunks of your skin off all the time. All right, if you keep going deeper, you move down into the hypodermis. That's where you have a lot of adipose tissue, fat, that's surrounded by areolar loose connective tissue. And if you keep going down deeper, you'll learn more about fascia. There are some denser connective tissues down in here between the hypodermis and the muscle. But just keep in mind there are more connective tissue layers down in here. Then finally, you're going to hit the skeletal muscle in most places if you keep going. And what do you have deep to the skeletal muscles? In most places, in many places, you're going to hit bone. Those are osseous types of tissue. So just kind of have a visual for the different types of tissues that you're going to be passing through as you go from the surface to deeper parts of the body. All right, skin structure and function. Be sure you can identify, of course this is for the anatomy lab test, all these different structures you see on a skin chunk and what they do, what are their functions. That's mainly going to be tested for you on the Unit 2 physiology exam. If you haven't taken the Unit 2 anatomy test, go back and review everything on that sheet, because you're going to have to identify these structures. It'll tell you, whether or not you need to know the tissue types that make them up. All of your major layers and sublayers, you got to be very careful about the terminology. Is it asking you about a cell type? Is it asking you about a sublayer? Is it asking you about a layer of tissue? So you want to be sure you go back and review all of those types of things. A good example up here in the epidermis, if there was an arrow pointing right here in the epidermis and it says what's the main cell type that you would find at this arrow. So cell type is going to be the keratinocyte. That's the main type of cell that you see in there, because those cells are busy producing keratin, which is going to end up here at the superficial part of the epidermis. But if it said what's the main tissue type in here, the tissue type is stratified squamous epithelium. That's a tissue. So you got to be careful about whether it's asking you about a cell type or a tissue. Or what if it said what sublayer are we in right here? Now generally I wouldn't ask for the sublayer on kind of a broad overview diagram like this, but you may realize that right in here, this is where the stratum spinosum sublayer of the epidermis is located. So go back and review all of those things. Down here in the dermis, if it said, okay, right in here in this little pink area, we can't see a cell, but what's the main cell type in there? Fibroblast. What major skin layer am I in? Dermis. What sublayer am I in? We're in the reticular sublayer of the dermis. All the pink stuff that you see there, what is that actually representing? If you guys remember, what's occupying most of that space there? You got your collagen fibers, you got your elastic fibers, and some reticular fibers, and well, that's responsible for that kind of pink coloration that you see there on the diagram. All right, then skin structure and function questions. Which of the following secrete substances onto the surface of the skin? Okay, so this is one of those multiple answer questions. How about the sebaceous glands? Yes, they secrete the sebum or your skin oil. Eccrine sweat glands. Yes, they secrete sweat onto the surface of the skin. Does the stratum lucidum secrete anything? No, the stratum lucidum, remember, occurs in thick skin, and that's a little thin layer of cells that have actually died during their journey on the way upward through the epidermis, and they're not really secreting or producing anything at that point. Arrector pili, those are the little smooth muscles in the dermis that pull on the hair follicles and give you chill bumps. They're not secreting anything. Apocrine sweat glands, those are your sweat glands in like armpits, groin area that produce the thicker sweat that's more hormone driven, anxiety driven. So that is a secretion, so they are secreting things. Meissner's corpuscles, Remember those? Those are the little delicate touch sensors that are up at the very top of the dermis. And so, no, they're not secreting anything. They're there for sensory reasons, sensing touch. Which of the following help protect the body from infection by bacteria and other microbes? What's helping protect us from infection? All right, so let's go through these. How about the sebum skin oil? Now, you might think no, but actually, if you recall, sebum has a little bit of a lower pH that we typically find in the body, and that lower pH of your skin oil is hostile to a lot of microorganisms. Not all of them. You've got plenty of microorganisms that live on your skin, but the sebum helps keep those under control. How about adipose tissues? Now, your fatty tissues aren't really protecting you from infection. Keratinized barrier of the stratum corneum, that uppermost sublayer of the epidermis. Yes, that's one of the functions of keratin. It's forming a barrier there that helps keep microorganisms, germs, out of the deeper parts of the body. Sweat. Sweat does have some substances in it that help protect you from infection by microorganisms. You will learn more about those when you get to Biology 202 and in your microbiology class. How about the corpuscles, like Pacinian corpuscles and Meissner's? No, those are sensory nervous tissues. Tightly connected cells of the epidermis. Yes, actually, that's one of the things that helps keep microorganisms from getting deeper into the body. The epidermis has tightly packed cells. And you'll learn in microbiology that's true for other areas inside the body where you have epithelial tissues as well, where they're all jam-packed tightly together. That helps keep microorganisms, bad guys, germs, from getting into deeper parts of the body where they are not supposed to be. Okay, the main reason healthy skin does not tear easily when pulled in any direction. So you can't, hopefully you can't just pinch your skin and rip off a chunk of it. That would be pretty bad. There are some people out there, though, who have a genetic defect that causes that to happen. All right, tearing easily when pulled. Pulling, think tensile strength. What component of tissues gives tensile strength? Hopefully you're thinking collagen fibers. Elastic fibers are for rebound. Collagen fibers help resist pulling. So your answer here is going to be C. Collagen fibers in the extracellular matrix of the dermis run in many directions and have tensile strength. So know your functions of these different skin and tissue components. Pressure applied to the skin. So I'm pressing on the skin. How do I feel, have that pressure sensation? What's responsible for those? Yeah, hopefully you're answering these, the Pacinian corpuscles. They kind of look like, on the diagrams, they look like yellow thumbprints or footballs, and they're sitting down kind of deep in the dermis, or down in the upper part of the hypodermis. So those are nervous tissues. When somebody's pressing on your skin, those Pacinian corpuscles react to that. They send a signal to your brain so that you feel that pressure sensation. Okay, and you certainly want to make sure you review the epidermal sublayers and their functions. Hey, my little mouse here is misbehaving. If I can get it to work properly. There we go. All right, so we're zooming in on the epidermis, and so we're seeing a little chunk of a papilla in the dermis down here, colored in pink. I do want to caution you guys, that doesn't mean your dermis is really pink like that. They're coloring it that way because when you look at skin slides in a laboratory, they are stained with colored dyes, and typically there's a purple dye and a pink dye, and the extracellular matrix of the dermis, the collagen fibers, the elastic fibers, take up that pink dye, and so that's why a lot of times they draw it that way on diagrams. All right, remember, your epidermis is divided into several sublayers. Down here at the very bottom, bordering the dermis, you have the stratum basale. And this is where you have cells that are dividing, undergoing mitosis, and then those new cells get pushed upward, 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 upward on their journey upward through the epidermis. So that's your stratum basale or basale. There are different pronunciations for that. Stratum spinosum, that is most, kind of the bulk of the epidermis in here. And spinosum means spindly. The cells kind of look like spindles, and they're busy producing keratin. If you keep moving upward, you hit the stratum granulosum, and the cells are getting really flat, and they're full of granules, and those granules contain developing glycolipids. If you guys, those are sugar lipid combinations that kind of help produce the waterproofing barrier that you have up here at the surface. And they've accumulated these keratin fibers that have been built. And then up here at the top you have your stratum corneum, and these are all dead cells, still filled with lots and lots of keratin that's allowing those barrier properties to occur. And then you've got those glycolipids, those sugar lipid combinations kind of fill in the spaces in between, and that's going to help us out with the waterproofing. And in thick skin, you have another sublayer in here, right in here. That's your stratum lucidum, where when you look at the cells under the microscope, they just look clear. They don't take up any of the dyes that they generally apply to those tissues, but you typically see that in thicker tissue like palm of the skin or the sole of the feet. Then some other cell types in here, dendritic cells, the little spidery looking purple cell there, that is, its job is to intercept microorganisms that are trying to invade through the epidermis. They have all these little arms that reach out and latch onto those things. And we spent more time talking about the melanocytes, like you see here. And they produce melanin, that's skin pigment that actually gets transferred out of the melanocytes and into the surrounding keratinocytes. They pick up that melanin. And do review your function of melanin as well and what its job is. All right, question: in which part or parts of the epidermis would you find dividing mitotic cells? Select all that apply. Where, all right, and you got to read carefully, in which part or parts of the epidermis would you find dividing mitotic cells? Select all that apply. Stratum corneum, hopefully you're saying no, that's just a bunch of dead cells full of keratin and glycolipids. Papillary sublayer, you may have some dividing cells in there, but that's not part of the epidermis, so we're not going to pick that one. Stratum lucidum, no, dead cells. The stratum basale, yes, that's your mitotic sublayer of the epidermis. The cells in there are dividing, and those daughter cells get pushed upward, upward, upward through the epidermis. Reticular sublayer, no, that's part of the dermis. It's not part of the epidermis. So be very, very careful with your reading. Most students need to slow down when they're taking tests. They're not reading carefully. So if I'm grading your test and Blackboard tells me you took 15 minutes or 20 minutes to take it, that's letting me know you probably were rushing through it and didn't have time or didn't take the time to read carefully. So you got to kind of get into the habit of slowing down and being very, very cautious about the wording, because almost all this terminology is likely new for you. So when you're working with new terminology, you got to kind of slow down so that you're thinking about what you're reading and making sure you understand what you're reading before you jump in and answer the question. All right, what is the most critical function of melanin? The most critical function: to give skin and hair their color. Yeah, it does that. To shield the DNA of epidermal cells from damaging UV light. Oh yes, definitely, that's a function. To help produce vitamin D? No. To provide a waterproof barrier? No, that's the function of keratin and not melanin. All right, so A and B are both functions of melanin, but what is the most critical? And hopefully you're thinking B, to protect the cells from the damaging ultraviolet light of the sun. Rule of nines on burns, that's something you need to review, and that's where different sections of the body have been divided up into areas that represent 9% of the surface area of the skin, or some multiple of 9, or some fraction of 9. And so you certainly need to review all of those things. The perineum area down around the genitals and the anus consists of 1%, so it's not really following that rule of nines. So let's see if we can apply the rule of nines to a question like this. A house fire victim has third-degree burns covering the anterior and posterior portions of both legs, the anterior portion of her left arm, and approximately 1/2 of the anterior side of her trunk. What percentage of her body would you estimate has been burned? All right, so let's think about this. Third-degree burns covering the anterior and posterior portions of both legs. Okay, so just one or the other would be 9% on one leg. If it was just the anterior side of the left leg, that would be 9%. But we have anterior and posterior portions of both legs. All right, so that's 9 plus 9 plus 9 plus 9. That's 36. We're up to 36. 1/2 of the anterior side of her trunk, anterior portion of her left arm. All right, if you go back, that's 4.5% of your skin surface. So we're now up to 40.5. And approximately 1/2 of the anterior side of her trunk. Anterior side of the trunk represents 18%. So if about half of that is burned, that's 9. So we're at what, 49, about 49.5% or so. So I probably should have made a 47 to 50% there to make sure we had that covered properly. There's 9 front and back, that's 36. Half of this is 9 more, that's 45, and then plus the 4.5% over here will give us 49.5. Okay, which of the following statements is not true about severe burn victims? So we had a burn case study available for you to work on. Answers should be discussed by students, not for a grade, but just for making sure you're prepared and understand the material in this unit. Please discuss these burns case study questions. So which of the following is not true about severe burn victims? Let's just go through these. The patient will lose water by evaporation from interstitial spaces, and this will draw water out of the bloodstream to replace it. Well, we got to do some thinking there. That's getting into what we've learned about body fluids and the movement of water. All right, so if you have a severe burn, your epidermis and your dermis have been destroyed. You no longer have that keratin waterproofing layer at the surface. So absolutely, water is going to be evaporating out of the interstitial spaces. And water moves from compartment to compartment. So if you're losing water from interstitial spaces, water is going to move out of the bloodstream, out of the blood vessels, and into those interstitial spaces to replace it. And so absolutely, that's gonna happen. The most critical need is pain treatment. Okay, a burn victim is going to be in severe pain, but is that the most critical need for the patient? Let's keep that in mind for a second. How about C? Loss of the keratinized epidermis allows entry of microorganisms into underlying tissues. Absolutely. That's one of the key functions of the keratinized epidermis, is to protect against infection. D, scar tissue will likely replace burned areas and restrict movement due to presence of excess fibers. Oh yes, in a burn victim, when these tissues get recreated, or even after some kind of just any kind of major injury, replacement tissues, when scar tissues form, it means the tissue's been replaced, but a lot of times the composition of the fibers is inappropriate. They tend to be too fibrous, and that will restrict your movements. And in that Burns case study, there's an example of that with the patient in that case study that you guys are working on. All right, so which of these is not true? B, the most critical need is pain treatment. Pain is horrible. Burn victims are in pain, but that's not the most critical need. What is the most critical need early? Hopefully you're thinking replacement of fluids. That fluid loss is the thing that will kill you right away after a burn, because you'll dehydrate. What happens to your blood pressure? Your blood pressure, as you lose fluids, is going to plummet, and if you can't circulate blood due to low blood pressure, your major organs are going to shut down and you're going to die. The next most urgent thing is infection. So it's fluid loss, infection. It takes a little while for an infection to get started, so that's not going to kill you right away after a burn. It's that fluid loss. That's the most pressing thing right out of the gate. All right, so those were just some example questions from Unit 2. Hopefully that's helpful to you. And I will post this PowerPoint that I used in the announcement for this, so you'll have that available if you want to flip through those questions and use those to help you study as well. And if you have any other questions that come up while you're preparing for the Unit 2 exams, please, you can course message me. I would prefer, again, that you post these on Piazza, the discussion board, so that everybody can benefit from them. So good luck finishing up your homework, and good luck with the Unit 2 exams.